In this video, we will discuss the prokaryotic replication fork and its associated portions and enzymes. Each strand has a 5' and a 3' end. DNA strands are antiparallel, so this 5' end must match with the 3' end of the other strand. The origin of replication indicates where replication occurs. Prokaryotes have many origins, but one is drawn here in purple. On the bottom strand, we see a zoomed-in image displaying the phosphodiester linkage. This linkage connects the 5' carbon of one nucleotide to the 3' hydroxyl of the next nucleotide via phosphate to create the sugar phosphate backbone. At the origin of replication, the DNA is melted and separated into single strands. Helicase is responsible for attaching to the replication fork and breaking the hydrogen bonds that exist between the two strands of DNA. The separation of the strands is necessary for replication of each of the strands to be able to occur. Helicase is shown in lime green. Gyrase is an enzyme upstream of the replication fork that surrounds the double-stranded DNA and relieves supercoiling that results due to the unwinding and separation of DNA strands. Gyrase is shown here in blue. Also shown on this image are the single-stranded binding proteins seen in orange. They're responsible for ensuring that the DNA is single-stranded and protected and stabilized. DNA has a tendency to resort to double-stranded state, which would result in hairpins in this case. As stated earlier, replication occurs bidirectionally, but now we will zoom in on one side of the replication fork to further examine the next steps. Replication can occur continuously or discontinuously depending on the orientation of the original DNA strand. Replication always occurs from the 5' to the 3' direction. The upper strand is the leading strand because the direction of replication is towards the right, in the same direction as the movement of the replication fork, which allows for continuous replication to occur. The bottom strand, on the other hand, runs from 5' to 3'. Since replication only occurs from 5' to 3', this lagging strand must be replicated discontinuously to form Okazaki fragments. DNA polymerase III is responsible for the addition of nucleotides to the growing strand. DNA polymerase III requires a 3' hydroxyl to be present in order to add nucleotides. The 3' hydroxyl comes from a primer created by RNA primase that is approximately 10 to 12 nucleotides long for the DNA polymerase III to add onto. A shortened primer is shown in purple, with the red being the nucleotides added by DNA polymerase III. RNA primase is shown in gray. Since the lagging strand replicates discontinuously, Okazaki fragments are created. DNA polymerase I can then come in and bind to the 3' end of the Okazaki fragments to remove the RNA primer and fill in the gap. After DNA polymerase I has done its job, there will still be nicks present in the replicated strand, indicating gaps that exist in the sugar phosphate backbone. Ligase can then come in and seal these nicks in order to form a complete and replicated strand.